Next organ is spleen. The spleen, situated high in the left abdominal cavity, is a large ovoid secondary lymphoid organ that plays a major role in mounting immune response to antigens in the bloodstream. Whereas lymph nodes are specialized for trapping antigen from local tissues, the spleen specializes in filtering blood and trapping blood-borne antigens, thus it can respond to systemic infections. Unlike the lymph nodes, the spleen is not supplied by lymphatic vessels. Instead, blood-borne antigens and lymphocytes are carried into the spleen through the splenic artery. Experiments with radioactively labeled lymphocytes show that more recirculating lymphocytes pass daily through the spleen than through all the lymph nodes combined. So that shows the importance of spleen. The spleen is surrounded by a capsule from which a number of projections or trabeculae extend into the interior to form a compartmentalized structure. The compartments are of two types, the red pulp and the white pulp, which are separated by a diffuse marginal zone. The splenic red pulp consists of a network of sinusoids populated by macrophages, numerous red blood cells, and a few lymphocytes. It is the site where old and defective red blood cells are destroyed and removed. Many of the macrophages within the red pulp contain engulfed red blood cells or iron containing pigments from degraded hemoglobin. The splenic white pulp surrounds the branches of the splenic artery, forming a periarteriolar lymphoid sheath. Pulse, populated mainly by T lymphocytes. Primary lymphoid follicles are attached to the pulse. These follicles are rich in B cells and some of them contain germinal centers. The marginal zone peripheral to the pulse is populated by lymphocytes and macrophages. Blood-borne antigens and lymphocytes enter the spleen through the splenic artery, which empties into the marginal zone. In the marginal zone, antigen is trapped by dendritic cells which carry it into the pulse. So lymphocytes in the blood also enter sinus in the marginal zone and migrate into the pulse. The initial activation of B and T cells take place in T cell-rich pulse. Here, dendritic cells capture antigen and present it combined with class 2 MHC molecules to Th cells. Once activated, these Th cells can then activate B cells. The activated B cells, together with some Th cells, then migrate to primary follicles in the marginal zone. On antigenic challenge, these primary follicles develop into characteristic secondary follicles containing germinal centers like those in the lymph nodes. For rapidly dividing B cells and plasma cells are surrounded by dense clusters of concentrically arranged lymphocytes. The severity of the consequences if the spleen is lost depends on the age at which splenectomy takes place. In children, splenectomy often leads to an increased incidence of bacterial sepsis. Next, cutaneous associated lymphoid tissue. The skin is an important anatomic barrier to the external environment. It is the largest organ in the body and plays an important role in non-specific or innate defenses. The epidermal outer layer of the skin is composed largely of specialized epithelial cells called keratinocytes. These cells secrete a number of cytokines that may function to induce a local inflammatory reaction. Scattered among the epithelial cell matrix of the epidermis are Langerhans cells, a type of dendritic cell which internalize antigen by phagocytosis or endocytosis. As mentioned earlier, these Langerhans cells can undergo maturation and migrate from the epidermis to regional lymph nodes where they function as potent activators of naive Th cells. In addition to Langerhans cells, the epidermis also contains so-called intraepidermal lymphocytes, which are mostly T cells. Some immunologists believe that they play a role in compacting antigens that enter through the skin, a function for which they are well positioned. The underlying dermal layers of the skin also contain scattered T cells and macrophages. Most of these dermal T cells appear to be ever previously activated cells of memory cells. Lymphoid organs and organ evolutionary comparisons. Spectrum of earliest vertebrates in jawless fishes, Aenata, to the birds and mammals, it is apparent that evolution has added a new organ of immunity, such as lymph nodes and tissue pairs, patches, but has intended to retain those. The thymus, for example, evolved by earlier orders, whereas all vertebrates have gut-associated lymphoid tissue, and most have some versions of spleen and thymus. Not all have lymph nodes, and many do not generate lymphocytes in bone marrow. The difference seen at the level of organs and tissues are also reflected at the cellular level. So far, neither T or B lymphocytes nor other components of the adaptive immune system have been found in the jawless fishes. In fact, only jawed vertebrates or Gnathostomata, of which the cartilaginous fishes, sharks, rays, are the earliest example, have B and T lymphocytes and support adaptive immune response. So now let's have a quick summary. The humoral antibody and cell-mediated response of the immune system results from the coordinated activities of many types of cells, organs, and tissues found throughout the body. Many of the body's tissues, cells, and organs arise from the different stem cell populations. Leukocytes develop from a pluripotent hematopoietic stem cell during a highly regulated process called hematopoiesis. Apoptosis, a type of programmed cell death, is a key factor in regulating the levels of hematopoietic and other cell populations. There are three types of lymphoid cells: B cells, T cells, and natural killer cells. Only B and T cells are members of clonal populations distinguished by antigen receptors of unique specificity. B cells synthesize and display membrane antibody, and T cells synthesize and display T cell receptors. So, most NK cells do not synthesize antigen-specific receptors, although a small subpopulation of this group, NK T cells, do synthesize and display a T cell receptor. So, macrophages and neutrophils are specialized for the phagocytosis and degradation of antigens. Macrophages also have the capacity to present antigen to T cells. Immature forms of dendritic cells have the capability to capture antigen in one location, undergo maturation, and migrate to another location where they present antigen to T cells. Dendritic cells are the major populations of antigen-presenting cells. Primary lymphoid organs are the sites where lymphocytes develop and mature. T cells arise in the bone marrow and develop in the thymus. In humans and mice, B cells arise and develop in bone marrow. Second, lymphoid organs provide sites where lymphocytes encounter antigen, become activated, and undergo clonal expansion and differentiation into effector cells. Vertebrate orders differ greatly in the kinds of lymphoid organs, tissues, and cells they possess. The most primitive and jawless fishes lack B and T cells and cannot mount adaptive immune responses. Jawed vertebrates have B and T cells, have adaptive immunity, and display an increasing variety of lymphoid tissues.